The pharmaceutical industry operates on a global scale. ICH Guidelines. Harmonized standards by ICH, International Council for Harmonization, under QSM, Quality, Safety, Efficacy, Multidisciplinary, to ensure global consistency in drug development and regulation. This necessitates a harmonized approach to ensure the safety, efficacy, and quality of medicines worldwide. This is where the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, or ICH, plays a crucial role. Established in 1990, ICH brings together regulatory authorities and the pharmaceutical industry from major regions across the globe. The goal? To harmonize technical requirements for pharmaceutical product registration. This collaborative effort streamlines the global development and registration process for new medicines. It also prevents duplication of clinical trials and reduces the need for redundant testing. The ICH develops guidelines that provide a unified framework for pharmaceutical development and manufacturing. These guidelines are not legally binding on their own. However, they carry significant weight and are often adopted into national regulations by ICH member countries. This harmonization simplifies the regulatory landscape for pharmaceutical companies. It facilitates international trade and, most importantly, ensures the delivery of safe, effective, and high-quality medicines to patients worldwide. ICH Q6, titled Specifications, Test Procedures and Acceptance Criteria for New Drug Substances and New Drug Products. Chemical substances stands as a cornerstone of pharmaceutical quality control. This guideline provides a comprehensive framework for setting specifications. Q-Series, Quality, covers pharmaceutical quality systems, including OQ8, Pharmaceutical Development, emphasizes quality by design, QBD, integrating scientific understanding into product development to ensure consistent quality, defines critical quality attributes, CQAs, and design space. OQ9, Quality Risk Management, promotes risk-based approaches to identify, assess, and mitigate quality risks, enhancing decision-making and resource. These specifications are essentially a set of criteria that a drug substance or drug product must meet to ensure its quality, safety, and efficacy. They act as a benchmark against which the quality of the product is assessed throughout its life cycle. ICH Q6 emphasizes a science-based approach to setting specifications. This involves a thorough understanding of the drug substance or product, its manufacturing process, and its intended use. The guideline outlines two primary types of tests: universal tests and specific tests. Universal tests are applicable to all drug substances and products. These tests include identification, assay, and impurities. Specific tests, on the other hand, are tailored to the individual characteristics of the drug substance or product. This may include tests for chiral purity, particle size distribution, or dissolution. The acceptance criteria defined in ICH Q6 are equally crucial. These criteria set the limits for various quality attributes, ensuring that the drug substance or product consistently meets the required quality standards. Non-compliance with these criteria can lead to serious consequences. These consequences may include batch rejection, regulatory action, or even patient harm. Therefore, adherence to ICH Q6 is not merely a regulatory obligation. It is a fundamental aspect of ensuring the quality, safety, and efficacy of pharmaceuticals. Impurities in pharmaceutical products can arise from various sources. These sources include the manufacturing process, degradation of the drug substance, or excipient interactions. These impurities, even in trace amounts, can potentially impact the safety and efficacy of the drug product. Recognizing this, ICH Q3, titled Impurities in New Drug Substances, and its companion guideline, ICH Q3b R2, Impurities in New Drug Products, provide a framework for the identification, qualification, and control of impurities. ICH Q3 mandates the identification of all impurities present in a new drug substance above a certain threshold. This involves sophisticated analytical techniques to elucidate the chemical structure and properties of each impurity. Once identified, impurities are then subjected to qualification studies. These studies aim to determine the toxicity profile of the impurity and assess its potential impact on the safety and efficacy of the drug product. Based on the toxicity data, acceptable limits for each impurity are established, ensuring that the final drug product is safe for human consumption. Q6&Q3 emphasizes a risk-based approach to impurity control. For more details, contact Priyadarshini College of Pharmacy, Koratagiri. This means that the stringency of control measures is commensurate with the potential risk posed by the impurity. Impurities with known toxicity profiles and those present above a certain threshold require more stringent control than those with low toxicity and low levels. This risk-based approach ensures that resources are allocated effectively to mitigate the most critical impurities, safeguarding patient safety without unnecessarily burdening the manufacturing process.